Thank you, everyone, for having me. I want to encourage you today to try to leverage pre-trained transformer models for protein structure or function prediction. I'm not a biochemist, but I do computationally study proteins. I'm a third-year PhD at North Carolina A&T, Aggie Pride. And for me, I generate novel protein sequences, but then I find new ways to evaluate them. My research has levered, or at least garnered, collaborations with Johnson & Johnson, Eli Lilly, and MIT Lincoln Lab. So one of the things that I've learned as a computationalist who does not have a background in biology is that there is a secret war going on. I call it protein wars. You have the good side and you have the sneaky / bad side. For those of you who do not know, we have antibodies, enzymes, hormones in our bodies to help us build different parts, repair, help us to fight off the bacteria and viruses that might enter, and just tell our bodies what to do. On the bad or sneaky side, we do have viral-based proteins that can come in and act as spies and trick our cells into letting them in. We do have rogue proteins that essentially allow diseases to form, and then we have misfolded proteins, which essentially can cause damage to different parts of our bodies. I would say that we've entered the twilight zone, for those of you who know, and we've experienced this with COVID-19 a handful of years ago when it shut the world down. It's not a protein, but it is a virus. It uses proteins in many different ways to help it shape, to assemble and release, to connect to the viral RNA genome, as well as attach to and enter human cells in many different ways. So AI has entered the chat, and one of the biggest advancements in the biochemistry space is AlphaFold. I believe we are now at AlphaFold 3. Essentially, it has streamlined the process of predicting a 3D structure of a protein sequence, something that could take months or even years to do, or a full dissertation to do. Now you can do it in a matter of hours or even minutes. Traditionally, it's an X-ray crystallography process that is usually in place. Then there are transformers, not necessarily these, but this one, the attention-based architecture. But before then, we had a neural network called Recurrent, and essentially it took in language, but one, let's say, segment or sequence or word at a time. So think like you're talking to a toddler and they're processing a word at a time versus talking to, say, like a high schooler or a college student. They can take a full body of text and understand that at once and communicate that back efficiently. So if you check out the paper, Attention is All You Need, you can find out more about that architecture. So the application of AI in the biospace, essentially it has dramatically changed how we, one, evaluate proteins, look at their function, etc. Experimentation has been streamed down, testing and drug development. We all saw that with the emergence of the COVID vaccines, something that typically takes 10 to 20 years to get through the door. For some, took less than a year. With that being said, one of the things that I, again, want to tickle your fancy about is just getting you interested in helping out in this space. So in terms of protein production, it is a natural biological process, but the thing is that we don't 100% understand all of their functions. And in fact, even the ones that are publicly available on the databases, they have not fully been experimentally validated. So we need researchers, we need people who are just curious to go out and help us do this computationally. With that being said, everybody is different. So we know that medications, food, our environment impacts us all differently. And so even though we might discover a protein, we may not know how it functions or reacts to different things. And because of that, there's more to study. There's a thing called dark proteins where we may know about it, but we may not know how it functions. And it's anticipated to be more superbugs like COVID-19 that comes to pass. And so we need those curious parties to help us to kind of figure out what these might be. With that being said, that help would help with essentially creating new drugs, creating new foods, creating new ways to basically help us to sustain our lifestyle here on the planet. In terms of two of the models that I want to bring to your attention, evolutionary scale modeling, which is more of an encoder-based transformer. It does have decoder-based properties, and what I mean by decoder is that it has the ability to generate. I believe that has been the fad here. ProtGPT2 is built on GPT-2, is decoder-based, so it does generate new protein sequences. With that being said, the lift to use these tools is pretty straightforward. I think you may have heard of Hugging Face throughout the day. You can pretty much use this through the Python package, or again, get you a quick token from Hugging Face. Essentially, for ProtGPT2 to start generating sequences, all you need to do is provide it with a seed, so a string of amino acids, and then it essentially generate new amino acids to that sequence. Or you can unconditionally generate sequences, which is what my research is focused on, and just ask it to generate new sequences. You can then take that sequence, put it through ESM, and try to figure out, okay, what is the likelihood of it to function in X way? So if we think that it might be an enzyme, we can put it through ESM to kind of figure that out. You know, enzymes, they help with speeding up a lot of processes in the body. And so if you find out that something is so novel, does a certain function, then it can help you with, oh, okay, this could be harmful, or this could be used for medications, etc. Again, this is something that is used, or at least the pre-trained version is on Hugging Face. There are other use cases with ESM. You can compare it to other proteins, you can look at patterns and other functional properties. So I hope that has interested you enough for you to start getting curious about proteins and maybe even leveraging some of these models that are out there. As part of my research, I am building some, so if you do sign up, you can go ahead and send me a connection on LinkedIn. I do have some reference papers and content. And then if you want to play around with these with a low bar, feel free to sign up. Thank you.
Thank you.